and welcome to our Spotlight on Careers event, exploring careers in science and technology. Today, we're joined by Dr. Volker Gerdts, Director and CEO of the Vaccine and Infectious Disease Organization International Vaccine Center, VIDO-InterVac in Saskatoon, located at the University of Saskatchewan campus. Dr. Volker Gerdts received a DVM in 1994 from Hanover Veterinary School and a PhD from the Federal Research Institute for Animal Health, Island of Reims, and Hanover Veterinary School in Germany in 1997. He spent 1998 to 2000 at VIDO-InterVac in vaccine discovery and mucosal immunology, doing a postdoctoral fellowship funded by the German Research Council. In 2002, Dr. Gerdts joined VIDO-InterVac as a research scientist. He was appointed Associate Director of Research in 2007 and became the Director and CEO in 2019. Dr. Gerdts' research interests are in neonatal vaccines for humans and animals, mucosal immunology, and vaccine delivery and formulation. His current research projects focus on vaccine development for porcine reproductive and respiratory syndrome virus, adjuvants, porcine immunology, and pigs' use as a model for humans' immune modulation and regulation of mucosal immune responses. So today you will hear from Dr. Volker Gerdts about the work that VIDO-InterVac is doing to create a vaccine for COVID-19. Please welcome Dr. Volker Gerdts.
When dealing with emerging diseases, you have to be really quick. And that is really something that VIDO is so good at. We're a very nimble organization. We quickly respond. When COVID-19 came, we were the first in Canada to isolate the virus, the first to have an animal model established, and the first to have a vaccine in testing. We're now manufacturing the material to be able to then go into what is called the tox study to show that the vaccine is completely safe. And as soon as we get green light, we will start our clinical trials in the fall to work with humans. And as part of the COVID-19 response now, we have lots and lots of interactions within Canada. We have, over the last few months, completed more than 40 studies on testing other people's vaccines. And we're also part of the World Health Organization expert groups. So lots and lots of international collaboration. And to be honest, if you want to be fast, nimble, and amongst the best, you have to work with others. Well, good afternoon. It's great to be with you. Thank you so much for allowing me to come and talk to you about what we're doing here at VIDO-InterVac and how a career in science might look like. What I'd like to do, as you've seen in the introduction, I'd like you to tell a little bit about the work that's happening here at VIDO-InterVac. We're very fortunate to have in Saskatoon one of the most advanced research organizations, organizations that are focused on the development of vaccines for both animals and humans. And so I'd like you to take a look at some of the slides that I have prepared, showing you what's happening here at VIDO-InterVac and how we actually make a vaccine for COVID-19. But as you've heard in the introduction, this is really about you, and so I'm happy to interrupt at any given time if you have any questions. At the end, I'm happy to have a Q&A where you can ask as many questions as you want. And if there's ever any question that you'd like to follow up on, my email is on the first slide. So you can feel free, or you can directly email me or go through your teacher, whatever works best. And I'd be happy to try to address any questions you might have. So let me just see if I can share my screen here. And if one of you could just confirm that you can see it.
We are good to go.
All right. So this is what we're going to talk about, the race against the clock, the development of vaccines for COVID-19. And down here, you can see our website, and I lied to you. I'm sorry about that. My email is not on here. This is the Twitter handle that you can go to, but you find me on our website. So if you'd like to reach out and contact me, you can just use this website here and then get in touch with me that way. So as I mentioned, I'm very privileged to work in one of Canada's largest research organizations for the development of vaccines. And so right here in Saskatoon is actually where we have one of the largest high containment labs in the world. And so this is what we need to work with these dangerous pathogens like the COVID-19 virus, but also others like the tuberculosis bacterium or any other viruses and bacteria that potentially can cause harm to the individual, even death to the individual. And so these facilities are specifically designed to work with these dangerous pathogens. And I'll show you how we protect our staff when they work with them. And so what you can see here on the right side, this is the glass wing. This is where the laboratories are, and some laboratories up here. And then over on this side, there is our animal facility. And in this line of research, we unfortunately have to use animals because you can't really mimic the immune system in any other way other than actually using animals for research. But you know, when we do these animal trials, when we do use animals in research, we are making sure that these animals are not suffering from any pain or any extensive manipulations or any of that. And all of our research is ethically approved by an independent committee that is hosted here at the University of Saskatchewan. So all of our research is ethically approved and okay. Now here's how our staff works with these pathogens. So you can see they're wearing these suits, these Tyvek suits. So they're essentially wearing the underwear underneath, and then they have a layer of surgical gowns on them, and then they wear these suits over. And what is unique is that on top they're wearing these PAPR units here. So there is a respiratory unit at the back that they carry with them, and that has a HEPA filter in it. And so the air that goes up is filtered, and it goes and blows over their head, away from their face, and then it comes out underneath here. And so in that way, they only breathe in filtered air, clean air, so they never have any chance of breathing in any pathogens that, for whatever reason, might be in the laboratory. However, we don't really work with these pathogens on the bench. Where we work with them is in these biosafety cabinets, as you can see here. So this is really something where you can see there's a tube, maybe there's the virus in there right now. So they reach in with their hands and they work within this biosafety cabinet. And here in front, they have this glass shield that protects them from any spills or any like that. And then going from here down, there is a curtain, an air curtain that comes. So there's air blowing out and it's being sucked into here. And so you reach through this air curtain with your hand. But what it does, it keeps all the pathogens in the inside of this biosafety cabinet. So really our staff never comes in direct contact with the pathogen other than here with their hands and the gloves that they're wearing. Now within this facility, we can house a lot of different animals. And this helps us to make vaccines both for animals as well as for humans. So sometimes you're working on vaccines specifically for cattle, for example, or for pigs, or for poultry. And sometimes we even work on vaccines like this one here for bison. So bison, for example, have tuberculosis and brucellosis, two bacterial diseases that are a big threat not only to the bison, but also to other animals like cattle, for example, that are being farmed or ranched at the same sites where the bisons are walking around. And so you often have spillover from the bison into the cattle population, and that causes them problems. But you can see we also work with bats, for example. We work with ferrets, as we will tell you, or as I will tell you today, or also with small rodents like hamsters, mice, and so on. And when COVID-19 hit, when our pandemic started early on this year in January, we put a large team of individuals together, many of them that are working directly in the lab or working as animal technicians in the animal side of it. We have people that are working on our contracts, on our administration, and so on. You can see a large effort to find a solution for COVID-19. And so VIDO-InterVac was the first in Canada to be able to isolate the virus in Canada. This was done from a patient sample in Ontario, and we were then we received the shipment here to Saskatoon, and we were able to isolate the virus, and then we shared it back with other laboratories, provincial labs, and so on in Canada to use the virus now for all the diagnostic work and so on. We were the first in Canada to have an animal model established, and as I will point out later on, that is very important when we want to test if a vaccine works or not. And we were also then within weeks, one of the first in Canada to have a vaccine candidate in development. It took us actually 5 weeks to develop it in the lab and have it in animals and start the testing ready. We're a member of a number of international organizations and expert groups, including the World Health Organization. And much of our work actually at the moment is also designed as Canada's research center to help others in developing their technologies, whether those are vaccines, antivirals, or therapeutics. So how do we actually develop vaccines? And so before we start with, I think this is the time for the first poll question. So question number 1.
All right, we have posted our first poll question. Will you get a COVID-19 vaccine? All right, so I think most of our participants have given their votes. So we have 75% said yes and 25% said maybe. We had 0 no's. So we'll see, Dr. Gerdts, if you can change their opinion in the next 45 minutes.
All right, so this is very, very encouraging. I'm glad to hear that 75% of you are actually looking or are willing to get a vaccine. That's higher than, as you know, what the national average here in Canada is. So when we make a vaccine, I guess the first question we always ask is, what is it that causes this disease? And so here's a very simple scheme. I'm sure you have this in your classes. So in this case, COVID-19 is caused by a virus. And viruses are these really, really small pathogens. As you can see there, they only have a micrometer in size. That's about half the size of a bacterium, as you can see below, and many, many, many times smaller than these insects, these ticks, for example, as you can see there, that we can see with our eyes. So what it means is we can actually not see viruses with our eyes. And if you would put them on the rim of a coin, on the edge of a coin there, a regular virus or even a larger one, you could put more than 10,000 viruses just on the edge here if you line them all up. That's how small they are. But still, they're very, very powerful. They can cause terrible disease, as we're seeing right now. And they can also transmit very easily. And here are ways of how these viruses can be transmitted. Because they are so small, you can see they can be transmitted with these very, very small droplets that leave our mouth when we cough, for example, as shown here in this picture, or they can make their way out with other body fluids, as shown here, in diseased or in contagious individuals. And so this is why it is so important for us to protect ourselves when we have these new diseases from these viruses. And one of the best ways, of course, is to clean surfaces and so on, but also to wear masks. So let's hear from you. How many of you are wearing masks outside of school?
The votes are starting to come in. We have almost everyone voted. All right, so 88% of our attendees said that they are wearing a mask outside of school, and the other 12% said no.
All right, so we're very clear. Some agree with it, the majority agree with it, but others don't. So these viruses, as I try to explain here, they travel very easily. And so what they do then is when we breathe them in, is they actually make their way into the upper respiratory tract. And so this is what I'm trying to show here. When we breathe now in what somebody might have just coughed out there, not wearing a mask, and you can see here's your respiratory tract. All of this in red is the mucosal lining. So mucosal is it's the surfaces that are covered with mucus, and that's why we call them mucosal surfaces. And so they're part of our respiratory tract, they're part of our intestinal tract, our genital tract, our eyes, ears, and so on. So all these surfaces that are covered with the mucus on top to make it smoother, that's what we call the mucosal surfaces. And so when you look at the respiratory tract, so into somebody's nose here, you can see all this in red is actually in layer of cells that are lined up right next to each other, as you can see on the slide here. So these are the nuclei in the center of the cells here. And there's one cell, here's another one, here's another, and so on. So they're lying right next to each other. And there's really only one layer of these cells that separates the airways, so the outside, from the tissue, the inside, the body. And so this is now where the coronaviruses are coming in. And this is where they're trying to get into these cells. And so viruses are pathogens that infect cells. And they need to get into the cell to then hijack the cell and make more copies of themselves. And so here is just a schematic showing how this works. So here's the virus on the outside, and the virus is trying to get into the cell, trying to inject its genetic code into the cell. And so what it needs for this is a molecule that it can attach to, so it binds to the cell surface. And when that is happening, now it can start to inject its genetic material into the cell. And then the poor cell doesn't know that this is not its own material. It just makes it like everything else, translates it into what we call proteins. I'm sure you've learned this in school. And then at the end, you can see now out of these proteins that the cell makes, new viruses are being assembled, which then are being released. And so one virus getting into the cell could potentially have multiple thousands of viruses coming out of the same cell. And so that is the problem, that when people are contagious, they often shed so much virus, much more than they actually were infected themselves. But the point here is that we need to have these receptors on the surface of the cell for the virus to attach. And when we now look at how this really looks, here now you can see the cell that I just showed you before in the mucosal layer there. And all of these are the viruses. These are the SARS-CoV-2 viruses that are trying to attach to the cell and make the way into the inside. And that's what we want to block or prevent with our vaccines. When they get in though, that's when we then start seeing problems. So this cell for a while produces multiple new viruses, but at one point it starts to die as a result of the infection. And when we have many, many cells dying, especially if it spreads to other sites in the organism, the liver, the kidney, and other sites, and then that's when we're seeing disease, and that's when we're starting to see the clinical problems that are associated with the disease. So let me ask you in the next poll question, how many of you personally, so not in the internet, but how many of you personally know a person that has had COVID-19?
Our responses are in, and 25% personally know someone who has had COVID-19, and 75% said no, they do not personally know someone who has had COVID-19.
So then let me ask you, how many of you, of those 25%, how many of you know someone who has gotten seriously ill from the disease?
All right, 88% said no, they do not know someone. And I'm not sure how this math adds up, but 13% said yes, they know someone who has gotten seriously ill.
Interesting. So this is really where it's great to live in a city like Saskatoon. Now you can see the vast majority of us have not met anybody, or we don't know anyone who seriously got ill from the disease, and that's fantastic. That's how it should be. If we were in larger cities or even in the US, the numbers would be very, very different. As you know, the caseload in Saskatoon compared to other places in the world remains very, very low. All right, so here's now the virus that I wanted to show you. So we talked about it, how it makes its way to the cell surface, and the protein, the structure on its surface that it uses to bind to the cell is what is called the spike protein. And it's called spike because it looks like a spike. It sticks out from the surface, and the reason for that is, as I showed in my previous slide, it needs to bind to the receptor right here. And so this is the protein that the virus uses to adhere to the cell. And this is what is also being seen by the immune system first. And that's why most of the immune responses we're making to this virus are actually directed to this yellow protein there, the spike glycoprotein. When I say yellow, of course, it's not yellow in the virus, it's just yellow in this cartoon here. And to be correct, actually, there's three of them together here. So we call that a trimer, when three proteins are coming together, and only if all three together can the virus adhere to the cell. Now, here is what we think how it works. The virus, as shown here, so here's SARS-2, here's SARS-1. So very, very closely related viruses. They use this receptor on the cell surface, and when they can bind to it, they get into the cell, as I showed you before, and that's when trouble starts. With vaccines, we're trying to block this from happening. We're trying to block the virus from adhering to the cell. And you can see here, antibodies are one of the possible mechanisms how this can be happening, how we can block this. And so antibodies are molecules that are made by our immune cells, and they're used to prevent, for example, viruses from binding to cells, and in that case we call them neutralizing, because they neutralize the effect that the virus might have. Now, if we look now at the vaccines that are currently in development and that you see on social media or in the news, there's the front runners. Some of them are even in phase 3 trials, and I explain what that means. But all of these vaccines focus either on the yellow protein, the spike protein directly, or they use what is shown here. When we use the whole virus, they have the protein in that vaccine mix. So we can make a vaccine that is essentially the whole virus, and we can inactivate it chemically so that we don't get sick when we get vaccinated. And now our immune system recognizes all of these structures here and makes an immune response to all of them. Or we can just focus on a subunit of the virus, a part of the virus, and most vaccines focus on the spike protein here. And so now we can either use the genetic material that encodes for this protein, so a small piece of the genetic code, and in this case, that would be an RNA vaccine or a DNA vaccine. And a prominent example that I'm sure you heard in the news about is the Moderna vaccine in the US. They're using an RNA vaccine, or the Pfizer vaccine, for example. It's a piece of RNA and just a piece that encodes for this protein. Another way of doing this is that we actually take the protein. And so this is what we here at VIDO-InterVac do. We are making this protein and then we're formulating it with what we call an adjuvant to enhance the immune response to it. So we're not using the RNA, we're using actually the product, the protein. And I'll show you over my next few slides how we do this. And then there is those vaccines that also use the genetic material, but instead of directly injecting the RNA, they use a vector, another virus that delivers this genetic material. And so often adenoviruses are being used. These are harmless viruses. Many of us have been exposed to them. Common cold, sometimes not even clinical symptoms at all. So they're very safe to use. And by inserting that gene that encodes for the trimer, you're now making a recombinant vector, a vector that carries now in its genetic code also the information for the spike protein of the coronavirus. So now you have an adenovirus that expresses some bits and pieces of a coronavirus. And that, a prominent example here is, for example, the Oxford vaccine, or the vaccine from Janssen & Janssen that is currently developing also another adenovirus vaccine. So how do we make these vaccines? Well, we start with designing them. And let me talk about that. But before we do that, let's hear from you guys. How many of you trust vaccines? Oh, we are with the safety right now. Okay. So the question is maybe not exactly the one that we wanted, but that's okay. How many of you feel confident about the safety of the vaccine? So let's see and hear what you think.
Sorry, I launched the wrong one, but we can go back to the other one. So right now we have 78% feel confident about the safety of vaccines and 22% say maybe.
Okay.
And we can go to our...
Can we go to, do you trust vaccines?
Yes. Do you trust the vaccines is launched, and we'll see what our participants say. All right, so 78% said yes, they trust vaccines, 22% said maybe, and 0% said no.
Wow, excellent. Okay, so let's see how we make vaccines. And as I mentioned, the first thing we start is we design them. So how do we actually design them? Well, we start with the genetic code. And so if you remember, this virus was discovered early, well, late last year, actually, early in January was then when the information came out, and the genetic code, so the sequence of the virus, was first published on January 12th. So these viruses, as I mentioned, have the RNA in it. So RNA, it's you know, as you know, is essentially a code. There's all these different nucleotides, and they encode them for proteins, or amino acids first, and proteins, and so on. And so if you remember what we just talked about, so this would be now focusing on the gene that encodes for this protein of interest that we want to make a vaccine for. And so the reason is that we want all these antibodies to recognize it. So this is how a sequence looks like, and this is not exactly the sequence for the spike protein, but you can see there is our genetic code, and you know this hopefully from your classes. There's these nucleotides, and it's essentially just a sequence of A, C, T, C, A, blah, 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 and so on, and it goes on forever. So we can use this then, and we have programs that can work with this information, and we can then design or learn how these proteins, what the sequence of them is, how they look like. So here's our genetic code, and if you remember, it gets translated into amino acids. These amino acids then together come together and form a peptide, which then in larger peptides make up these proteins. And so this, in this case here, would be now our spike protein of the coronavirus. So we have now the information, we have the genetic code, and we know how the protein needs to look like. And that's what we need to include our vaccine. And so how we do this is we make use of these little factories that now actually make the vaccine for us. We don't do this ourselves, we don't synthesize it chemically. What we're using is cells, culture cells that make this for us. So all we have to do is now introduce the genetic code that we just saw, that we got in early January, into the DNA of the cell, and then the cell, as a little factory, makes for us the protein that we're interested in. And it even is so nice to secrete it into the supernatant. So all we have to do is now harvest it from the supernatant. And so our job as a researcher is to do this, put it in, and keep these guys happy. So now every cell can make many different proteins secreted in this supernatant. And so that we actually, when we grow these cells now in these plastic flasks, as you can see here, on the bottom of this plastic, you have a layer of cells. There's your individual cells. You can see them how they look like. And now they carry this gene that we're interested in, the spike protein. And they produce all this protein into the supernatant, the red fluid that you can see here. And that's where we're harvesting it from. And so the way we do it for vaccines is that we don't use these small plastic flasks or just for the early phases, as you can see here. Later on, we use some very large bioreactors that have up to 2,000 liters in there. And in there, they have billions of cells. So billions of little factories that, if they're happy, are making the protein for us. And that's how we make a vaccine. These cells, these proteins are producing, these cells are producing the protein, the spike protein for us. And now we just need to harvest it, which is shown here. We do this in very clean, almost sterile facility using very complicated equipment, but we essentially purify it to the cleanest grade you can imagine, and then we stick it into these glass vials, put a cap on it, and there's our vaccine. Now that is only one part of the vaccine, and that is what we call the antigen, and that's what you might find in some of these newspaper stories. That is the part of the virus that we're interested in. That's what we want the immune system to recognize and make an immune response to. We also formulate it then with adjuvants, and these are molecules that just tell our immune system, come on, let's go now, make an immune response. So we're enhancing the immune response, and that's if you had ever a shot that kind of stung a little bit and hurt and maybe even had some swelling and so on. That is often a reaction of the adjuvant, in that we purposely want to activate the immune system so that our vaccine is being recognized by the immune system, all the information is being taken up, and we actually start to make a good immune response. We also then put some preservatives and so on in there to make sure the vaccine doesn't go bad when we store it and when we ship it around the world, especially when we ship it to communities that are hard to get to and so on. And these are all parts of it. Now, instead of using these little factories, these cells, you can also use plants, and that is shown on the right side. So the same idea here, we put our genetic information into these plant cells, and now the plant is growing, and when it's growing, it's making more of our protein of interest. We have to then find a way of getting the protein out of these plants, but technically that is actually not very difficult. And as you can imagine, we can grow lots and lots of plants. So this is another very effective way of making vaccines. This is, for example, what the Canadian company Medicago out of Quebec is doing right now. And they have a COVID-19 vaccine that is already in clinical trials in humans. Or you go to the left side where you have this ugly soup there. And that ugly soup, if you would look into this, is full of bacteria. So rather than using little factories like mammalian cells or culture cells, you can use bacteria to do the same for you, and then you harvest the protein from this bacteria. So these are different ways of how we can manufacture a vaccine, but the principle is always the same. So how do we actually know now that the vaccine works? Let's hear what you guys think. Question number 6.
Before we go to that question, Dr. Gerdts, we had a question in the chat.
Oh, sure.
Someone asked, because it's just the protein, is that why it doesn't make you sick? No
Yes.
Genetic material.
Yes. Good point. Thank you for bringing that up. See, I didn't even mention that in my talk. Exactly. That's why one protein alone cannot make you sick. So a subunit vaccine is very, very safe. Excellent question. So let's hear now, how do we know that a vaccine works?
All right, our voting is in progress. Do you think the vaccine might work? And we have 88% said yes. Oh, it just went down. 78% said yes, 22% said no, said maybe, and 0% said no.
Interesting. Well, that's good to know. So let me explain then how we actually test whether the vaccine works and what we need to do. So here's how we normally make these vaccines, and you can see the number here of importance. It takes a long time normally to make a vaccine. And that is because in the initial phase, there's a lot of testing that is required. So what I'm going to show you in one slide, in fact, means months and months of work, and sometimes even years. But once you then have your prototype vaccine here, at this stage where it has shown efficacious, so it works in animals, then we can go into clinical trials. So we call this the clinical phase. And there we're looking initially for, is the vaccine safe for it to be used? And we use less than 100 volunteers in these studies. And then we go up to hundreds, so many hundreds of volunteers. And we also ask now, is it safe? And are we seeing the responses that we'd like to see? And only if this is a yes to both, then you are allowed to go into what is called a phase 3 trial, now involving thousands of volunteers. And here you ask the question, how does the vaccine perform out there in the field? So this is what's going on for some vaccines right now. They're testing them in thousands of individuals in the face of the disease, just out there, and then we'll see how the vaccine works. After that, you get the regulators to review the information, the data, and if everything looks good, you get then permission to start manufacturing. And so globally, we have already some that are in these phases here between phase 3. In Canada, our fastest right now, the Medicago vaccine is phase 1, and we're starting our phase 1 trial in December of this year. Now, so how do we know this? We use animals. And so again, as I mentioned, we don't like to use animals, but unfortunately, it's the only way of how we can do it, because we need to have an immune system that responds to the vaccine. And so here you can see ferrets, and they're being, in this case, they're exposed to the virus. And so there's other models you could also use. So hamsters is a good model for COVID-19, for example. And so are some of these non-human primates, which normally it's very rare to work with. Most work, of course, is happening with species like these ones. This is really the last step before a vaccine goes into humans. And so what we then look for in these animal studies is, do we find these antibodies that are described to you? Do they neutralize? Do we see T cells involved in this? Do we get protection from the disease? Does the vaccine actually work? How long does the immune response last? How many immunizations do we have to give? How much of each dose do we have to give to get good responses and so on? And what we are interested in overall is to determine how well does the vaccine work, how effective it is, how efficacious it is. So we're looking at vaccine efficacy here right now. And so there's lots and lots of work that goes into this, and this is very, very exciting. So if you wanted to, think about a career in science, this is absolutely fascinating, in that there is lots of really interesting things you can discover and that you have to address to come up with a vaccine. And so this is just an example how it may look like. Now we're looking at some antibodies in the blood of these animals, so in serum. So these animals just donated some blood. We collected it like you do when you go to the hospital. And then we measure how many antibodies we find in these antibodies. And it allows us then to compare different vaccine candidates, as you can see here. Now, the next question then, of course, is after we have determined it works well in the animal model, well, is it safe? Can we use it in humans? So let's hear from you guys. How many of you, and we had that already, I think, how many of you feel confident about the safety of the vaccine? Do we want to do that again, Michelle, then quickly?
I'm trying right now, but I don't think it's letting
If
me
not, then
launch.
I think the responses were, oh yeah, there we go.
There we go, we're good.
Excellent.
So has Dr. Gerdts convinced you of the safety of vaccines? Do you feel confident about the safety of vaccines? All right, it looks like most have voted. 89% said yes, 11% said maybe.
All right, well, it's a slight improvement. That's already fantastic. Good. So what we're doing in these trials, as you can see here, again, we're first testing the vaccine safety in animals, as you can see there. And at one point, then we're going into humans. These are volunteers that are participating in these trials. They get an injection like you can see there, and then they're being monitored for weeks after the immunization to see if there's any unwanted reaction to the vaccine. And so here is the phases that I described. So initially we go with 2,200. We start in an age group of 18 to 55 years old, and then we include older age groups and younger age groups as the data comes in and as the regulators are saying, okay, you're good to now go to another age group. And so what we're looking for is any unwanted reaction, either locally, so where we inject it, or systemically, overall, how our vaccine does make you feel. Do you have fever, any unwanted reactions, or any of that? Once a phase 1 trial is successfully completed, you can then go into phase 2. You can use hundreds of volunteers now. And again, you continue to look for any unwanted side effects. But you also are now asking, is there any immune responses? Are these individuals responding as our animals did in an earlier stage? Are we seeing the same, what we want to see? And then if the data is looking good, you can go into these hundreds of or thousands of volunteers, as I mentioned in phase 3. And that's really where we're saying then how well does the vaccine work? And so this is then if everything gets reviewed then by the regulators, eventually, you may get permission to start commercially manufacturing vaccines and make them available for the public. And so, so my next and my last question is really, how can we develop these vaccines better? And so this is really what we do here at VIDO-InterVac now a lot. With the current COVID-19 vaccine, we have seen that in a response to a new disease, we're already much faster than what we normally are. Normally, it takes about 10 to 15 years to make a vaccine. Now we're trying to do it in 1 to 2 years. But even then, as you will witness, and as I do, it affects our lives. It costs the economy trillions of dollars. Each of us is affected by the disease. Our lives are affected, our sports activities, our hockey games, all that is affected. And that is, we're trying to catch up to the disease. So a new disease emerges, and now we're starting to make a vaccine. So what we at VIDO-InterVac are doing is trying to think about how can we make vaccines before a new disease emerges. What do we need to know about any future pathogens so that we can make vaccines for it prior to them even emerging? So they're not even existent yet. And this is where really advances in bioinformatics, artificial intelligence, but also in structural biology are helping us now. We can predict what these new viruses might look like, where those mutations may occur, and how these mutations then affect the virus itself, the structure, and so on. And then we can use the various animal models and make these pathogens jump the species barrier, like they did for the SARS-CoV-2, and create a new virus, a new disease. And if we do this all in the lab, we know what a possible virus might look like, and we can make a vaccine. So that when this thing actually happens in real life, we could send out an airplane to wherever it happens in the world, organized by the World Health Organization, and drop off 10 million doses to be used right away. And even if this vaccine was not 100% effective, it still will help us to slow down the spread of the disease. So this is really where future research will help us all to control these pandemics much better. And this is where we need you to be part of this. This is something that the next generation's job is. You guys need to take this on, but I am convinced we will have vaccines for new diseases that will be available before we even actually have the disease. So with that, I'd like to move over to poll 8 and 9. Those are then the last two questions, but you know, our institute gets a lot of support from the government, and I'd like to recognize those in my last slide here. So do you trust the scientists developing the vaccines? Do you trust me? Let's hear.
All right, well, it looks like 89% said they trust you and 11% said maybe. They're not completely sold yet, so we'll see if your the rest of your presentation or your answers to the questions can have them convinced.
Right, so let's go to the social media one. Where do you get your information from?
So for this poll question, you can pick more than one. So let us know which of these sources you get your COVID-19 information from. So it looks like 67% said social media, 67% said news media, 22%, which is higher than this morning, said their peers, 33% said parents, and 33% said doctors and experts.
Interesting. All right, so do we have any questions in the chat there?
We do have a number of questions for you. So one question said, in the earlier slides, it stated that the clinical phase takes 10 to 15 years. Does it mean the vaccine being raced towards COVID-19 isn't safe enough as they are not adhering to the stated years of testing?
Right, so excellent question. So the difference between what we're doing now and what we normally do is we're trying to accelerate each and every step without cutting any corners. And so traditionally what we would do is first testing, look for the data, second activity testing, look for the data, next one, and so on. So it's all linear, and that's what takes a long time. Now what we're trying to do is trying to do as much of it as possible in parallel already. So while this is going on, we're starting already this activity and then this activity and so on. And so what it means is that if something doesn't work out, we may lose a lot of money. We wasted all of what we did here, but that's right now because of the urgency of the situation. People are willing to lose money, risk money on candidates that don't work. And so very early, we start with the manufacturing, for example, to then find out, well, maybe it didn't work, so we have to throw this out and focus on something else. So it's just it's the same process shrunk by doing things more in parallel. It's the same thorough process that you normally would have.
Our next question from a student says, what kind of undergraduate degree would help you get a job at VIDO-InterVac? Would a biochemistry major do?
That would be super if you had that, but there's also others, you know, biology, microbiology, immunology. We have all kind of different staff working here. Some have, like our technicians, for example, have undergraduate degrees from university, or even a SIEC degree, which is really, really good for working in the labs. If you have a PhD and you want to become a scientist and one day maybe even a professor, then you have to have a PhD in an area that's relevant. So biochemistry, microbiology, immunology, those are areas that for sure will help you to find a job here.
We have a question from a teacher, I believe. She said, in class, we have discussed the characteristics of life, and students had to do research to determine whether viruses are alive. Students in Lipton, Saskatchewan would like to know, are viruses alive?
So my definition would be no, but it's how you define it. So viruses are organisms that can get into a cell, so therefore they have the characteristics of being able to get into something, but they can't themselves reproduce unless they use the cell to do it for them.
We have another question saying, is it true that researchers need blood donors from COVID-19 survivors?
So we haven't had any here yet, but what it does allow you to test whether your vaccine is being recognized by the antibodies that are in the blood of these survivors. So patients that have survived an infection, their blood now has all these antibodies that help them to clear the infection. And so we're interested in seeing if these antibodies recognize, for example, our vaccine. So it's a very important step along the way to be used.
The next question is, if a vaccine is approved in another country first, will VIDO continue to work on their own vaccine, or will they start producing the approved one?
So we cannot produce the approved one unless we get a license to it, and we're not at the moment ready with our manufacturing facility. We only got funding this March, and so it's going to take, unfortunately, a year to produce it. And only would we be allowed to do that under a license from that company. We will continue to make our own vaccine. We think it's very important that as a country, Canada does not depend on other nations to make vaccines for Canadians. We believe that it is important to have a made in Canada vaccine that is eventually is available to Canadians. And this is not going to go away. This coronavirus, COVID-19, is going to be around for many, many years, and we will need, just like the flu shot, most likely every year an immunization for this one too.
How do you find people to participate in the human trials?
It's amazingly not a problem at all. We have had throughout the process, I would say, I don't know, we must have had hundreds of emails from people all across Canada early on in the spring who had volunteered to be part of our trials, people from Halifax to Vancouver, all over the country.
When you started your career journey, did you ever think you would become the CEO of a place like VIDO-InterVac?
So I didn't. I started, I went to vet school, but I always wanted to go into research. I went through vet school because I had heard it gives you a really, really good foundation to build on, and it does. It teaches you how organisms work, how our organs work, and you know, how it all works together. And so I was always fascinated by research, and what I wanted to do is work in a research institute. So I used my summers to contact institutes just like VIDO, but in Germany. I grew up in Germany, and I volunteered in these institutions, and I wanted to just see and learn what people are doing. And so then I got so excited about it that I was very fortunate to then do a PhD in immunology, and then the rest is history. I came over here at one point, went back to Germany, and then came back here, and just you know, kept working and being interested in what I do, and it all worked out at the end.
All right. Well, I think that brings us to the end of our list of questions we had from our participants. Thank you to those who asked questions. And thank you to Dr. Volker Gerdts, who's given up his time to share the work that VIDO is doing with COVID-19 research. And I know in our office here, we're all very excited to hear about this work as we hope to find the vaccine and be able to share that with our community in the near future so that we can have a life like we used to live.
Right.
Thank you for joining us today. It was great to hear from you. And before I log off, I have a little bit of information that I am going to share with our participants. So anyone who was viewing our presentation today, we asked that you would provide us with some feedback. So at the Saskatoon Industry-Education Council, we provide career exploration for students throughout the year. This month, we are focusing on careers in science and technology. And so we'd ask that you provide us with some feedback. So if you watched our presentation today, please let us know what you thought. We have a QR code for students to scan. And if not, if you don't have a QR code generator or you don't have your phone on you, we do have a website that you can go to give us feedback. And we love to give away prizes. So if you provide us with some feedback, you will be entered in a draw to win some gift cards. Teachers, if you were viewing this with your class today, we ask that you provide us with some feedback as well. We would love to know where you're located, which school you're at, and how many students and what courses you were watching this particular presentation in. So we have a teacher feedback form. Both of these feedback forms are very quick and it won't take you long, so we would greatly appreciate if you took a little bit of time to tell us what you thought. So once again, we have a QR code for teachers to scan, or we have a website that they can go to. And teachers, don't worry, you'll be entered to win the gift cards as well. And so with our Spotlight on Careers, every month we will be focusing on a different sector. So this month we looked at careers in science and technology. In November, we'll be looking at manufacturing and mining, December, healthcare, January, mental health and wellness, February, business, finance, and entrepreneurship. In March, emergency services, April, the skilled trades, and May, information and communication technology. We will put all of our live events on our website as recordings. So if you missed any of our live events, you can find them on our website. And you can also find other virtual resources available. We have interviews with industry professionals. We have resources for teachers and students, as well as classroom activities. And VIDO-InterVac was kind enough to create a lab activity for teachers to use in their classroom. And we also have other resources that were created from the CDC, as well as agriculture in the classroom, which can be related to science and technology careers. And finally, to stay up to date with the SIEC and know what we have going on throughout the year, follow us on Facebook, so Instagram or Twitter, or you can subscribe to our YouTube channel, which is where we will house all of the recordings from our live events. Thank you for joining us today, and we look forward to seeing you at our events in the future.